Hello and welcome to my channel. This video is my first reaction video to Dr. Jack Kruse, and he talks a lot about melanin, mitochondrial light, and deuterium. But he never really talks about liquid-liquid phase separation, and I think that's an important thing to understand when you're trying to understand how all these things are interconnected. And that's where most of my research has been done on liquid-liquid phase separation. So I wanted to make this video to talk about the things I agree with and the things that I think he needs to go a little bit further. So his main argument is that mitochondria don't simply make ATP, they also generate light, and he thinks that that light is used for signaling and energy generation due to the fact that melanin can interact with light. Also, light's also called electromagnetic radiation, and the isotope composition of hydrogen, known as deuterium, may influence mitochondrial function. So let's get right into it. There are a lot of papers written on the photon emission of mitochondria, and the current literature shows that mitochondria really can produce extremely small amounts of electromagnetic radiation known as ultra-weak photon emission, or UPE, due to the production of byproducts in the form of reactive oxygen species. When some of these molecules return to their ground state, they can release photons. However, I would separate the two statements that mitochondria produce photons, which is experimentally supported, and the fact that those photons form a major biological information network, which is what Dr. Kruse is implying, which is very intriguing, but it's just hypothetical. It's not really established. So if you want to read right here, you can see how part of the electron transport chain in the mitochondria produces reactive oxygen species, and whenever those reactive oxygen species go back to their ground state, they release photons. And those are just little packets of light. So there's also a lot of research on the fact that melanin can absorb light and interact with light. So melanin, and this paper shows eumelanin, which is just the brown form of melanin. There's different types of melanin, like neuromelanin, and these are the two chromosomes shown right here, the chromophores that are associated with melanin, and chromophores can interact with light due to these aromatic residues. So melanin really does have unusual photophysical properties and protects biological molecules from photochemical damage. Kruse's hypothesis describes melanin as something resembling a biological battery or energy transduction system, and has connected it to mitochondrial light and water chemistry. While that's a fascinating hypothesis, saying that melanin absorbs electromagnetic energy is very different from demonstrating that melanin provides a quantitatively significant source of usable metabolic energy comparable to the mitochondrial oxidative phosphorylation. So that second claim that Dr. Kruse is imposing, it requires more evidence. And you can see here that melanin does interact with a lot of different wavelengths, that's different colors of light. And whenever melanin does interact with this light, it does go through proton exchanges with the water around it. So it can also interact with the water around it. However, it has not been shown that this somehow generates ATP and is part of some uh, ATP energy production for cellular metabolism that outnumbers the ATP production from oxidative phosphorylation in the mitochondria. So that's something that, while is a fascinating hypothesis, needs to be looked at a little bit more. So let's talk about hydrogen a little bit. And Dr. Kruse talks about heavy water, also known as deuterium, and how it plays a significant role in health and disease. And while that's, again, a very intriguing hypothesis, science has only just begun to collect evidence on how that might work. So it's still in the hypothetical realm. So replacing ordinary hydrogen with deuterium can produce substantial isotope effects in biological systems. Research on deuterium depletion in human health exists, but reviews of the field emphasize that the evidence base remains relatively small and conflicting. So we're still trying to understand it. And one of the best papers I've found to support that deuterium has an effect on biological signal transduction is this paper on the androgen receptor. So this is the androgen receptor right here. You can see the different protein domains. Part of these protein domains are intrinsically disordered. It has been established that intrinsically disordered proteins in the right conditions can undergo liquid-liquid phase separation, which is when it aggregates into a different phase of water from the surrounding water. So you can see right here that both parts of the androgen receptor can go through liquid-liquid phase separation, and that when they introduce deuterium can modulate this process. So this is the best paper. You can read more by looking it up. This is the best paper showing that liquid-liquid phase separation can be modulated by deuterium. However, again, Dr. Kruse never mentions liquid-liquid phase separation. So if he really wants to get a consistent hypothesis from the quantum realm all the way to physiological function, he needs to try to gain a better understanding of what's going on in what's called the mesoscopic scale, which is how molecules form macromolecular structures like fibers and protein lattices. And while he talks about protein lattices every now and then, he always leaves out liquid-liquid phase separation, which is actually what determines the physical state of those lattices, whether they're aqueous or gelatinous or fibrous. So they can exist in different states. It's regulated by liquid-liquid phase separation, and that would connect the quantum to the physiological function. That's the middle bridge that would connect the two. And we'll get more on that in later slides. So let's continue. Kruse takes another step and attempts to connect these observations into a much larger theory of biological organization. I think we must distinguish between what has been demonstrated, what is plausible, and what remains speculative. Furthermore, I think there's an emerging area of biology that could provide part of the mechanistic framework missing from this discussion, and that's, as I said before, liquid-liquid phase separation. I do have two different papers that explain liquid-liquid phase separation in The Origin of Life and Evolution. And I also have a paper with the Taylor and Francis group called Chaos to Coherence, A Path to Molecular Organization, which explains the quantum effects of these phase-separated systems and how the quantum effects can organize the proteins associated with liquid-liquid phase separation using the water as a substrate. So the really interesting question is, why would cells produce photons? If endogenous photons have a biological function, what molecular system could receive and translate that information? Biology usually doesn't allow energy or information to simply float around without some mechanism of organization. Signals affect proteins, proteins change conformations, molecular interactions reorganize, signaling networks become spatially organized. And this is where I think liquid-liquid phase separation deserves much more attention. At the smallest scale, we have electrons, protons, photons, hydrogen bonds, and isotope effects. At a larger scale, we have proteins, DNA, RNA, membranes, and metabolic pathways. Liquid-liquid phase separation is one candidate for that intermediate organizational layer that connects these scales. Because phase separation can amplify relatively small molecular changes into much larger changes in cellular organization, a small change in protein-protein interaction strength can determine whether thousands of molecules remain dispersed or suddenly assemble into a condensate. So, what I mean by disperse or a condensate, you can think of these liquid-liquid phase separated biomolecular condensates of RNA, proteins, or DNA. You can think of how they organize together in water is similar to oil in water. So, in the same way, oil, which is hydrophobic, organizes into little droplets in water, in DNA, RNA, and proteins, in each of those macromolecular structures, you have certain molecules that are hydrophobic and certain molecules that are hydrophilic. So those hydrophobic molecules, those nucleic acids in DNA and RNA that are hydrophobic, or the amino acids that are hydrophobic in proteins, will organize together so that a bunch of proteins and nucleic acids will come together and form these little bitty droplets. And that's important because they have to have a way of organizing themselves within water even when there's no membrane. So membranes in organelles like the nucleus and the mitochondria and the membrane on the cell, those do a good job of organizing biomolecules like proteins and DNA, but you also need a way for those molecules to organize just in water itself. So how that happens is liquid-liquid phase separation through this mechanism where those hydrophobic portions cause those proteins and nucleic acids to aggregate together so they form these little droplets, which can create signaling networks where anything from ions to pH to sugar concentration to post-translational modifications and even light can have an effect on whether these condense together or dispersed back into a dilute phase of water. And if you want to read more on this, please read my paper titled Liquid-Liquid Phase Separation as a Driver of Abiogenesis and Evolution, because I explain this mechanism very well. It acts as an on and off switch of dispersed or condensed, and it follows a sigmoidal curve. So mitochondria themselves use phase separation, which is associated with many different diseases, as you can see here and read in this paper. And you can see that liquid-liquid phase separation plays an essential role to mitochondrial function in transcription, RNA post-transcriptional processing, and degradation. So it plays a role in many different processes in mitochondrial function. So a hypothetical mechanistic bridge and something that's testable is mitochondrial metabolism leads to redox state reactive oxygen species that produce light, proton chemistry, solvent environment, and then that leads to protein interaction energies. and then the protein interaction energies determine liquid-liquid phase separation and biomolecular condensates, and that determines cellular signaling and gene expression, and all of this together ultimately determines cellular phenotype, which is the morphology or structure of the cell. And as we know in biology, structure and function are correlated, so depending on what structure you have, it leads to some type of physiological function, like the difference between a fibroblast and a macrophage. That gives us actual molecular steps that can be tested experimentally. So we need to not jump to deuterium means disease, or mitochondrial photons, or definitely biological information. Instead of making that jump, we need to explain it mechanistically like this to come up with a testable framework. So could light interact with liquid-liquid phase separation? And besides this example that I give, there's also examples of photosynthesis. The photosynthetic systems in plants exhibit liquid-liquid phase separation that is mediated by light, is induced by light. So we already know experimentally that light can be engineered to control phase separation from photosynthesis and from optogenetic systems such as the optodroplets. They use light-sensitive protein domains to induce condensate formation. Modern optogenetic studies use this principle to modulate phase separation with precise spatial and temporal control. Here's a link to that paper if you want to see it and read it. That doesn't prove that naturally produced mitochondrial photons regulate liquid-liquid phase separation, but it establishes an important principle that photonic energy can alter molecular interactions strongly enough to control phase behavior when an appropriate molecular absorber is present, like aromatic residues. So a legitimate research question becomes, could naturally occurring chromophores, potentially including aromatic amino acids, melanins, and other molecules, couple with endogenously generated light to produce changes in condensate dynamics? We don't currently know, but unlike simply saying biophotons control biology, that's an experimentally testable hypothesis. So I agree with Kruse that modern biology can sometimes become so focused on individual genes and biochemical pathways that we forget cells are physical systems. Biology operates through chemistry, but chemistry operates through physics. Electrons move, protons move, photons interact with molecules, water reorganizes, proteins change conformations, and molecular assemblies undergo phase transitions. Kruse is trying to force people to think across those scales. I think that's very valuable, but I do think he's missing some important aspects at different scales of this mechanism, like liquid-liquid phase separation. And whenever you're one way to describe how all this works and is in my paper titled Chaos to Coherence: A Path to Molecular Organization, is instead of thinking of biology as DNA makes RNA and that makes proteins and that leads to function, you need to think of the whole system. So that's water molecules, ions, electromagnetic fields, hydrogen bonds. You need to think of all of that as a complex adaptive system. So for more, please read my paper. So where I think he goes a little too far is treating proposed connections as though the complete mechanism has already been established. For example, mitochondrial ultra-weak photon emission is real. That does not prove that mitochondrial photons form a major coherent communication network. Melanin has extraordinary photophysical properties. That does not prove melanin is a major source of metabolic energy in humans. Deuterium produces real isotope effects. That does not prove that most chronic diseases can be explained through deuterium accumulation. Those distinctions matter because it's science, because the most interesting ideas don't need to be exaggerated, they need to be tested. So an experiment that I would like to see if you were trying to prove this model so that we can learn how to modulate it in disease is you choose a model for liquid-liquid phase separation. There's many different models you could choose. You control the solvent isotope. You have different concentrations of deuterium and water. You modulate mitochondrial metabolic state, possibly by using those different concentrations of deuterium. You introduce light, so you can try to understand if light has an effect on mitochondrial metabolism or mitochondrial biomolecular condensate liquid-liquid phase separation. And you observe and quantify the liquid-liquid phase separation. And you can measure all these different variables here, like the size of the liquid-liquid phase separated droplets, the viscosity of the droplets, protein conformation, all of these variables. So you can integrate variables systematically and design a factorial or matrix experience where you vary the percent of deuterium, mitochondrial state, the wavelength of light or protein concentration, and what proteins you're using, or maybe fluorescently label proteins so you can see how they move in the mitochondrial matrix, or maybe in the mitochondrial membrane, wherever you want to see them move. And you can ask important questions like, does increasing deuterium shift the phase boundary? Does mitochondrial metabolism change liquid-liquid phase separation? Does light at specific wavelengths promote or disrupt liquid-liquid phase separation? Do these effects interact? Can the effects be predicted by measurable parameters? And the goal would be to determine whether isotope-dependent solvent dynamics and mitochondrial photochemical activity can modulate protein phase separation and consequently cellular signaling and gene expression. If yes, then this provides a mechanistic bridge from physics to chemistry to phase behavior and biology. And I actually do think that whenever you incorporate deuterium, you probably would see an increase in the phase separation in the mitochondria. However, does melanin play a role? Does the light produced from the byproducts of oxidative phosphorylation, does that light have an effect on liquid-liquid phase separation and ultimately lead to melanin acting as some sort of energy molecule? I'm not sure about all that. We need to test these things to gain a better understanding. So in conclusion, the missing link may be phase behavior from photons, isotopes, and metabolism to life. Liquid-liquid phase separation connects physics to physiology. Multiple real phenomena do occur, like mitochondria emit ultra-weak photons, melanin absorbs and transduces light, deuterium alters molecular dynamics. These phenomena are real, but jumping directly to disease or information skips essential layers. And that layer might be liquid-liquid phase separation at the mesoscale. So at the mesoscale, liquid-liquid phase separation could be a substrate where the modulation of quantum interactions like photons, electrons, protons, isotopes, and water structure could translate directly to physiological function, gene expression, metabolism, and different cellular phenotypes. And it gets that those quantum scale interactions translates to these physiological scales through liquid-liquid phase separation. So that's an important layer that we need to investigate, and that is currently being investigated. So again, a testable mechanistic chain is you have mitochondrial metabolism, redox state reactive oxygen species, and the generation of photons. Proton chemistry, solvent environment leads to protein interaction energies like in intrinsically disordered proteins. Those mediate liquid-liquid phase separation and biomolecular condensates. The structure of biomolecular condensates determines cellular signaling, gene expression, and ultimately cellular phenotype or cellular morphology and structure. Each step is measurable, each step is testable. Together, they form a mechanism. Phase behavior can amplify small physical changes into large biological outcomes. As we've said multiple times, you can look at this chart again if you'd like right here. By studying liquid-liquid phase separation as the linking layer, we can finally connect light, water, isotopes, and metabolism to biology through a testable, quantitative framework. The goal is not to accept or reject ideas, but to test the links. Liquid-liquid phase separation may be the hidden organizing principle that makes those links possible. From physics, chemistry, phase behavior, and to biology, we can build a science that unifies scales and reveals mechanisms, new mechanisms that we can understand and we can utilize to modulate disease states. But we must understand them. We must make a thorough story and mechanism at every scale. We shouldn't just jump from biophotons and different isotopes of water to disease states. We have to know the structure of water. We have to know how that structure of water changes signaling mechanisms and how that ultimately leads to cellular physiology and pathophysiology. And we can only do that through experimentation and hypothesis-driven research. So I thank Dr. Kruse, Dr. Jack Kruse, for coming up with these extraordinary hypotheses that he has gained throughout his life. And I hope we can continue to research them. And I hope that he looks at liquid-liquid phase separation, because I think that will help him introduce and explain these ideas to the world. So if you enjoyed this and you want more, please like and subscribe, and you have a wonderful evening.